All right, everyone, welcome. My name is Eric Kalachik. I am the director of Boston University's Hariri Institute for Computing, and I want to welcome you all virtually to the Institute today for our Distinguished Speaker Series. We're kicking off a new cluster of speakers today in the context of machine learning for model-rich problems. Let me just walk through a couple steps here. First, our program. I'll do a brief intro here, and then I'm going to turn it over to Allyson Sgro, who is one of our junior faculty fellows here at the Institute, and she's going to introduce our speaker. We're delighted to have Cecilia Clementi here to speak with us. She'll give a presentation for a bit over a half hour, and then we're going to leave some time at the end for some Q&A. As an important administrative housekeeping detail, if you want, we encourage you to participate and ask questions using the Q&A feature, and then the Q&A will help inform the discussion that we do afterwards, which Allyson will moderate. So if I can take just a couple minutes to tell you, those who may not know us, about the Institute. So the Hariri Institute for Computing at BU is dedicated to initiating research convergence and accelerating integrated initiatives with social impact, particularly when they're at the nexus of computational and data sciences. The Institute includes a number of centers and initiatives that work in collaboration to advance this ambitious research portfolio, and you see a variety of them listed there. We also, at the same time, represent a community of over 250 researchers in computing and data science across campus, doing amazing work in areas ranging from misinformation to cybersecurity to artificial intelligence, cloud computing, contact tracing, conservation, and more. This isn't what you came to hear, but this is just a tip of the iceberg of what's been done here. So let me tell you a bit about the series, and then I'm going to turn it over to Allyson. So the Distinguished Speaker Series here at the Hariri Institute is aimed at bringing innovative speakers with bold ideas and computing-enabled and data-driven areas of research to BU. The series hosts a handful of speakers clustered around a common theme over usually a 2 to 3 week period. Each speaker is asked to present individually, and then they again come together for a panel discussion at the end of the week. Right, and so today we have Professor Cecilia Clementi speaking. This will be followed over the next couple of weeks by talks from Miguel Besa, Jacob Bortnick, and Joshua Blumenstock talking on different areas, all of which bring machine learning and model-rich elements to bear. So I'm going to turn this over now to our host. Allyson Sgro is Assistant Professor in Biomedical Engineering and also a Junior Faculty Fellow at our Hariri Institute. She's an East Coast native here from the US who left high school early to attend Simon's Rock College of Bard, where she earned a BA in chemistry and pre-medical studies. She moved to the University of Washington, Seattle, where she developed new tools for biological analysis and obtained her Ph.D. in chemistry in 2011. From 2011 to 2016, she was an NIH Kirstein NRSA Fellow and a Burroughs Wellcome Fund CASI Fellow at Princeton, where she studied the origins of collective behavior in Dictyostelium. And then in January 2017, she joined BU as an assistant professor in biomedical engineering and a member of the Biological Design Center at BU. She later became a Moorman Simon Interdisciplinary Career Development Professor here, and she holds a joint appointment programmatically with the Department of Physics. So Allyson, I'm going to turn it over to you.
Thanks so much for the introduction, Eric. It's my pleasure today to introduce Cecilia Clementi as a fellow someone who focuses on data-driven biology. She's an Einstein Professor of Physics at Freie University in Berlin, where she started in June 2020, though I think working remotely as many of us have tried to during the pandemic. Cecilia's attained her Ph.D. in physics at the International School for Advanced Studies in Trieste, and then was a postdoctoral fellow at the University of California in San Diego, where she was part of the La Jolla Interfaces in Science program. She spent 19 years as a professor of chemistry at Rice University in Houston, Texas. And her research focuses on developing and applying methods for modeling complex biophysical processes, which is what she'll be talking about today, particularly focusing on molecular models for machine learning experimental data. But she also integrates molecular dynamics, physical mechanics, and coarse-grained models as well. Her research has been recognized with the NSF Career Award in 2004, as well as the Robert A. Welch Foundation's Norman Hackerman Award in Chemical Research 2009. And she's also the co-director of the National Science Foundation's Molecular Sciences Software Institute. So Cecilia, please take it away.
Thank you very much for the introduction and for the invitation. I feel really honored to start this new series. And I want to tell you a little, let me share my screen first of all. Do you see my screen?
Yes, now we do.
Okay, I'll try to tell you, since I don't have much time, I'll tell you one story. Something we have been doing. I've been interested in modeling molecular systems at reduced resolution in a coarse-grained fashion for a very long time. And now with machine learning, I think we really, this kind of research has really taken off powerfully to a new direction, because I hope I'll show you that using machine learning, we can enable science that we couldn't do before. First of all, I want to introduce the problem. Why are we interested in coarse graining? Molecular systems, biophysical systems, are characterized by a large range of interconnected length and time scale. And this is what makes them so difficult to study. In physics, usually in undergraduate or in graduate school, we are told that in order to solve a problem, you have to decide what is the length scale and the time scale characterizing the system, and then you find the tool for it. But when there is a large range of interconnected time and length scales, things are more complicated. It's a multi-physics kind of approach. So for instance, if you consider the smallest scale, the atomic scale, the molecular scale, like for instance, here I'm showing a water molecule. Water is very important for biology. If you consider just a molecule per se, a single molecule, a single small molecule, then you are interested in the electronic distribution around the nuclei, and then you use quantum mechanics and quantum chemistry to try to find the orbitals, the probability of the electrons, the location of the electrons around the nuclei. And that gives you all the chemistry information and the reactivity of the molecule and so on. But if you are at a little bit larger scale, for instance, if you take a larger molecule like a protein, this is a mid-sized protein, then now you have many atoms, and the larger scale is lower time scales, and then you cannot use quantum mechanics anymore. So if we use quantum mechanics in the, at the smallest possible scale, we need to use some coarsening procedure that is not necessarily just because we cannot use quantum mechanics. It's also because we don't care anymore about all the variables that we would care at the smallest scale. When we have a protein, we don't necessarily care about the position of the electrons around each single nuclei. We care about the global motion. So we take some averaging of the electron position, and we define a molecular mechanics force field, and this is what we do with all atom molecular dynamics. And with all atom molecular dynamics, we can study slower time scale, like nowadays we can go to microsecond or even millisecond with special hardware and software. And this allows us to study protein folding, protein conformational changes, and so on. But if we want to scale up to the full cell, then this is also not enough. Both because it's computationally too expensive if you go to such a large system, but also because it's too much information. We don't need the information about each single atom, the position of each single atom, when we consider the full cell. So you do something like a very coarse approach, and you do, for instance, reaction diffusion simulation, where one bit, one interaction unit is a full protein. So this is a very coarse model, and this is used, for instance, in full cell simulations. There are several groups doing this. What we are interested in my group is to try to bridge this resolution, do something in the middle, where we have large system or multi-molecular systems like big complexes with more than one large protein, that is not at the, we don't care about this, the fine resolution of the atomic resolution, but the coarsening that we do, or like one protein, one interaction center, is too much. We need something in the middle, and we would like to do it in a systematic way. We would like to be able to build tools to bridge this resolution, to do some model reduction in a systematic way. Also, there is the question, what is the appropriate resolution? That is an important question. We are working on this direction too, but I'm not going to talk about that today. So when we talk about finding intermediate resolution, we talk about coarse graining, defining coarse models. And in my view, coarse graining is similar to, is well illustrated by this series of pictures by Picasso. These are all pictures of a cow. These are all drawings of a cow. They all represent the same thing, but the resolution is different. You go from the very detailed drawing to the very schematic, just a sketch drawing. They all convey the same idea, but the level of detail is different. And depends, the right resolution is what depends on your goal, depends on what you want to convey, depends on what the problem you're trying to solve is. And the same thing is for molecules. There is not, in principle, one absolutely best resolution. It depends on the problem at hand. So we want to do the same thing. We want to define a series of models for a molecule at different resolution in a rigorous way. So this is coarse graining in structural. There are different kinds of coarse graining. You can do coarse graining in time, coarse graining in different spaces. This is coarse graining in the physical 3D space. It means that we group together atoms into some pseudo atom. We call them beads. And for instance, we can group together side chain in a protein. A side chain can be an interaction center and the backbone interaction center, or we can put more atoms together to go coarser and coarser. And of course, we want this to be physically meaningful. So we want these to reproduce some properties of the real system. We want to study, want to use this model to study the real system. So we need to do it in a way that is physically meaningful. And for this, first of all, we need to define, mathematically, we need to define a mapping, meaning that we need to define the resolution. We need to say, how do we go from the atomistic, from the atomic details to the coarse-grained representation, where several atoms are grouped together in one pseudo atom, an interaction center. And in the following, we're going to consider only linear mapping, meaning that the coordinates of this coarse-grained bead is always going to be a linear combination of the coordinates of the corresponding atoms. If we have like 10 atoms mapping into a bead, we substitute these 10 atoms with one interaction center. The coordinates of this interaction center are given by the linear combination of the coordinates of the single atoms. Or we can even take a linear combination, can be a special case of a linear combination, can be a slicing of the space. We say we take one atom as representative of 10 atoms, something like that. And this, when the problem of finding a good representation is also an interesting problem. And I said we're working on it, but I'm not going to talk about today. Today we assume that the representation is given. We say we want to work at this representation. We have this mapping. We go from this to this simplified view. Then the next step that is very important is to associate an energy function such that the property of the system, the thermodynamics, for instance, of this system reproduces something real. Because at the atomic scale, we have force fields that have been designed to reproduce experimental data and also from integrating from the quantum mechanical derivation of, like as I said, you can integrate out the position of the electrons that you obtain with quantum mechanical computations and have some rigorous first principle of energy for the atomistic representation. But in the coarse-grained approach, we don't have a first principle way to associate an energy to a bead. A bead is not a real atom, is a pseudo atom. So we need to find a procedure that gives us an energy that allows us to reproduce something physically meaningful. And there are different ways to do that. First of all, this is where why machine learning is so important, because this approach, either direction we take, and we're going to go through the possible directions in a second, we need to use a lot of data. The bottom-up approach means that we decide that our coarse-grained model is going to reproduce some properties as they are measured in the atomistic system. So we trust our atomistic model, we trust our atomistic force field. We want the coarse-grained to reproduce the same properties, for instance, the same thermodynamics. If we measure a free energy with the atomistic model, we want to measure the same free energy when we do simulation with the coarse-grained model. So it's some sort of bottom-up. You integrate out degrees of freedom, and you want to match some properties. Or you can take a top-down approach, meaning that you want to reproduce experiments, some global properties. For instance, you want to reproduce some average properties, some average thermodynamic property, not detailed free energy landscape, but just one number or a few numbers. And this is top-down. Or there are other ways where you use experimental determinate structure for proteins, for instance. So in my group, we use all these approaches. At the moment, we are pursuing all of them independently, but our goal is to combine everything together at the end in the future. I'm mostly going to tell you today about the bottom-up approach, because deep learning plays a very important role here. But if I have time, I'll also say something about the top-down approach. So one way people have been doing coarse-grained model, as I said, is first of all to fix the resolution. We take a protein and we decide what, for instance, in this case, we consider only the carbon alpha as representative of fuel residue. So a carbon alpha is one particular atom in an amino acid. It represents the full amino acid. So it represents like more than 10 atoms, about 10 or more atoms. So it's a pretty coarse representation. Also because usually these models integrate out all the water. Proteins work in water. So when you do all atom realistic simulation, you have to consider the water molecules too, the water molecules in the environment too, while coarse-grained model integrate out some of the atoms of the protein and also all the water, usually. There are also coarse-grained models with explicit water, but in what follows, we'll integrate out the water. And then you need to associate an energy function with a lot of parameters. And traditionally, people have been giving a functional form and adjust the parameter to, with the top-down or bottom-up approach, to reproduce some properties that we want to reproduce. And usually this energy function has some local steric terms like angle, bonds, dihedral, and some non-local interaction to mimic van der Waals or electrostatic, this kind of interaction. And usually these functional forms are given that are similar to the functional form in the atomistic representation. For instance, this one is a van der Waals, it's modified van der Waals interaction. That makes sense for the atomistic model because quantum mechanics tells us that the two atoms coming together behave, interact with each other with the potential that is the van der Waals potential. But we don't know if this makes sense for a coarse model because we don't have atoms anymore. We have normalized, we have renormalized some degrees of freedom out. So there is in principle no reason why we fix one functional form and adjust the parameter in a coarse-grained model. One particular way what we follow, what we'll use in the following to do coarse graining is to enforce thermodynamic consistency. What is thermodynamic consistency? Here I said that one way to do bottom-up coarse graining is to take a fundamental or atomistic description that can come from, in turn, from quantum mechanics, for instance, or we can start directly from quantum mechanical description, integrate out some degree of freedom, obtain the coarse model, and we require this coarse model, the way we design the energy function is for this coarse model to reproduce the same thermodynamics. That means the same free energy. When we project on the same collective coordinates, we measure a free energy that we can do with, if we have a simulation with statistical mechanics, we can obtain the free energy from simulations. And the free energy we compute in the atomistic representation must be the same as the free energy we obtain with the coarse model. This is a requirement of the model for the model to be correct. We call this thermodynamic consistency. And turns out that if this is what we want, there is only one and only one energy function possible for the coarse-grained model that is given by this set of equations. Because by the definition of probability and partition function and free energy, in order for the free energy to be the same, the energy of the coarse model, this U of X is the energy of the coarse model, is related to the energy of the atomistic model, that is V of R, when we integrate out the extra degrees of freedom. So it's simply like really the integrating out degrees of freedom. So this, and we can show that this potential, this energy function, this effective energy function for the coarse-grained model is unique. There is one and only one energy that satisfies thermodynamic consistency. So this seems easy, seems that the problem is solved. We want thermodynamic consistency. We want to define a coarse model to match the thermodynamic of a more detailed model. There is one energy that does the job once the resolution is fixed. The problem is that it's easy to write this equation, but in practice, this cannot be solved. These are extremely complicated integrals. These are very dimensional integrals. These are, if R is the coordinates of the atomistic representation, means that is the X, Y, Z of all the atoms of the protein in the water, can be thousands and thousands, tens of thousands of atoms. And so this is a very, very dimensional integral. It's impossible to do it either numerically or analytically. It's impossible. So this is explained here in a very easy case where it's possible to do the integral. If you go from 2D to 1D, you integrate out 1 degree of freedom. Let's say we want the same free energy of this system in 2D, or when we project it 1D, then we just integrate out the second dimension, that is the Y here, and we find the 1D potential, that is this one. When we run simulation on this potential, we obtain the same free energy in 1D as when we run simulation on this two-dimensional potential. We want to do the same for our protein, but we go from thousands and thousands of dimensions to one handful of dimensions, one handful of atoms that represent the full protein. But fortunately, that's been worked out by mainly by Will Noyd and Greg Vogt, that there is an alternative way to define this potential. This set of equations is equivalent to a variational approach. If we can consider all possible functions, the space of all possible functions, the function U that satisfies this thermodynamic consistency is also the function U that minimizes this functional, where F are the forces of the atomistic system, and they are projected into the coarse-grained system. It's not important to follow the details here. The important thing is that there is a variational approach that gives us as a minimum the potential that satisfies thermodynamic consistency. But the minimum is the absolute minimum in the space, is a functional, so is in the space spanned by all functional form that we cannot explore in practice, especially if we take a guess, if we can take an ansatz for the potential and we take a trial function for the potential, as is usually done in variational approaches with some parameters, then we restrict this minimization to a certain functional space, and there is no guarantee that the minimum corresponds to the minimum, the true potential that we would get through these equations. It's the closest possible, but maybe still far. And this is what has been, how this force matching variational principle has been used with the ansatz function and minimization of this force matching error. It's called force matching because there is a force entering here. Changing the parameters to minimize this functional form. One important thing here is that the minimum of this functional is never 0. It's always larger than 0. And the lower value, the lower possible value, is because of this renormalization. It's only due to the fact that we are renormalizing degrees of freedom. So this is a very noisy estimate in machine learning. It's a very noisy estimator in machine learning speech. So the fact that we have this functional gave us the idea that what hinders the progress here is the fact that we cannot explore the whole functional space. But this is what neural network is good at. Like for instance, neural network has been introduced as a universal function approximator. So if we use a neural network in U, if you use some machine learning way to approximate U instead of giving an ansatz for the function, we can go closer to the real potential, the potential that does the job, the coarse-grained potential that we want, that is thermodynamically consistent. And so this is what we did. We consider the data here, the input data are the forces for all atom simulations, from all atom simulations of a given system. And the output would be the potential U that satisfies thermodynamic consistency for a coarse-grained system. And what we do is we define a loss function where we minimize this object with the data, over the data. This is a functional, but we approximate it with data, with the data we have. And for you, we use a neural network, a deep neural network. And this is what we did a couple of years ago. We proposed what we call CGNet, where basically this free energy net is what I just said, is a neural network that approximates the energy of the coarse-grained model. As input, we have the coordinates of the coarse-grained model. As the output, we have the energy. So this is an energy function, but it's a complicated energy function where everything interacts with everything and is modeled by this neural network. There is a featurization that means that we transform the coordinate into a set of coordinates that are rototranslational invariants to enforce the physics, the symmetries of the system. And we use some prior energy. It means that we also enforce some part of the energy that we know must be there. For instance, the existence of bonds, the existence of excluded volume, two atoms cannot overlap in space. So all the physics constraints go into the prior energy, and the rest is modeled by this free energy network where everything can happen. We let the neural network decide what's interacting with what. We applied it to several systems. One interesting system is signaling, that is a very small protein that folds and unfolds. We coarse-grain it from the full system with all the atoms and water. And even if it's a small system of only 10 amino acids, this is already thousands of degrees of freedom. We go to 10 beads, only 10 pseudo atoms. So this is 30 degrees of freedom. We go from several thousand to 30 degrees of freedom, that XYZ for 10 beads. And it works very well. This is the reference. This is the free energy we want to match. This is as a function of two reaction coordinates. Different colors here represent different value of the free energy. Blue is deeper, so the minima in free energy are the most probable state. And you see there are 3 minima. That means there are 3 metastable states. One folded state, that is A, and you see this is the folded configuration. One unfolded state, that is B, where the molecule is open. And one misfolded state, that is C, where the protein is compact but not correctly folded. And if we use our CGNet model, we reproduce exactly the same, 3 deep minima. And if we extract structures from these minima, they are very similar to the corresponding atomistic structure. So the model does the job. Instead, if we use an ansatz, a only two-body interactions ansatz for the free energy function. Here, instead of a neural network, we put some functional form where we allow only pair of bits to interact. We don't get anything. We just get the blob in the free energy. So we don't have distinct minima. We don't have folding and unfolding. While if we use the neural network, we do. So already here we suspect that the trick is that the neural network allows multi-body interactions, 3 bodies, 4 bodies, and so on, that are very difficult to model otherwise. This was a proof of principle. More recently, we have been taking this to the next level, and because here we have used all-atom simulation to train a coarse-grained model. So the question is, if you already have the all-atom simulation, why train a coarse-grained model? You already have the free energy. You reproduce the same thing. You do all the job and you reproduce the same thing. The idea would be to train a neural network on small molecules like these, and then use it on very large molecules where a coarse-grained representation is more meaningful, and you cannot even do an all-atom representation because the system is too big, and do it at a different level of coarse-graining. This is what we introduced last year in the SGSnet. I don't want to go into detail. This is the neural network architecture. And the main difference is that the featurization, the transformation of coordinate from Cartesian to these features to represent the translational rotational invariance and permutational invariance coordinates is learned by the network. And also the network is composable, meaning that it's transferable. The architecture is transferable. If we put together a sequence of amino acids, the architecture of the network is composable in parts that represent the different pieces in the sequence. So if we change the sequence, we can just reshuffle these parts. So this is transferable. We can learn it in a system and apply it in another system. And indeed it works. We again used it on signaling, and it works even better. This figure is not so clear, but it works even better than was working before. And this is because this architecture, forcing the transferability, regularize the system. It imposes some constraints that makes the energy smoother. And this is more clear in this figure when you see we measure the error between, the error is the difference between these two free energy landscapes. It's not easy to measure distances in high-dimensional spaces. So we defined different measures, like we can measure the difference between the equilibrium probability distribution of the system using the Kullback-Leibler divergence, or the mean square error of these two-dimensional surfaces. And in both cases, the error we make with the SGSnet, with this transferable architecture, is smaller than the error you make when you use the ad hoc architecture only for that system. So this is very encouraging. Now we are applying to a very large set of protein to train all of them together to have some sort of universal, or at least for a family of protein, force field, coarse-grained force field that works on all of them and is transferable to other proteins. And here again, for SGSnet, we get ensembles of configurations that are very similar to the ensemble that we get in the all-atom representation for the 3 minima of the system. And this is a movie I want to show you. This is the movie produced by the neural network. So if you use the neural network as an energy and run a molecular dynamics simulations, instead of the energy, we just put this network. Input is coordinates, output is energy. So we can use it as an energy function, although it's a neural network. It's a function, but it's complicated. And if we run simulations, this is what you see in the free energy landscape. This is what you see as in the folding reaction coordinate. And there is a collective transition. Now it folds, and you see the structure gets to the folded state. So it does the job. It folds and folds as it would do for the real system. But the energy function is learned by the neural network. Now, I said that we suspected here that when we compared to a two-body energy function, that the trick is done by the multi-body interactions, because the difference between what we call the spline model and the neural network model is the fact that the neural network can very easily model multi-body terms. Everything interacts with everything in your neural network. So we decided to understand this better, and this is actually now in press. I just returned the proof of this paper, so it should appear soon on JCP. We did what in physics is called the multi-body expansion. We designed a different architecture where we had, first we do a two-body net, when only two bodies interactions are allowed to exist. Then we add a three-body interaction. Then we add four-body interaction, and so on. And each of these terms is modeled with the neural network, meaning that we don't restrict ourselves to a specific functional form, but the neural network can span a larger, more expressivity in the functional space. And what we find is very surprising is that, yes, indeed, multi-body interactions are very important, but they are decaying. This multi-body expansion decays very slowly. In particular, here, if you focus on the KL divergence, the Kullback-Leibler divergence measure of error, you see the two-body makes a very large error with respect to the CGNet is what we compare with. That is what works, what we know works. But even just adding three bodies is not enough. Adding four bodies is not enough. Here we add four bodies and chirality. And then we add five bodies, and indeed we find something comparable to CGNet. Usually, when you do this multi-body expansion in physics problem, you hope to have only like a three-body as a perturbation of the two-body. But here, the expansion decays very slowly. So this really, for me, underscores that at the moment, unless we understand better what's going on here, we need to stick to machine learning. We need to use machine learning for the representation of the energy of the system, because we don't have another way to model all desired body interaction with functional forms. Hopefully, neural network and machine learning will also allow us to learn something about the physics of this interaction, so that we can perhaps approximate this interaction with some functional form, and then we can do the traditional approach, like molecular dynamics with the force field. But at the moment, I think if we want to do coarse graining in a rigorous way, we need to use machine learning. And there are a lot of data, a lot of molecular dynamic simulations of all atom systems that are available that we can use for training. And this again is a picture showing the same thing. Here is the reference, the all atom. And this is how the free energy landscape changes. So this is the stand where we want to get that. This is when we have two bodies, and we already know that it doesn't work. Three bodies starts to work. At least there is a little bit of separation between these states. Four bodies works better, but it's still not perfect. Four body and chirality is better, and five body becomes even better than what we started from. So five body is really, really similar to the reference. Also, you see it in this measure of errors here. So we also think that this multi-body decomposition works as a regularization again. So by constraining this interaction to a certain number of bodies, we impose a smoother energy, and so we get closer to the reference system, to what we consider the ground truth, that is the atomistic simulation. This is, and now, as I said, we're working on training on multiple proteins to have a really some sort of universal force field that works on at least a family of proteins altogether, not just on one protein. This was for the bottom-up approach. As I mentioned, we also do top-down. I see that I don't have much time. I just want to mention that what is top-down means that instead of defining a model, then doing the simulation, then comparing to experiments through statistical mechanics. We do the reverse. We start from the experiments and we define a model to reproduce the experiment. And because eventually, really what we want is to reproduce experiment. So we need to have a way to incorporate experimental data into this model. And there is a lot of experimental data we can get information from. And we are doing this with the maximum likelihood approach. Basically, we modify the energy function of the coarse model by defining a likelihood of the model to represent the experiment and maximizing this likelihood. I don't have time to go through all the results, but again, I want to show you the final thing. Using this approach, we have been able to represent, for instance, use NMR data, and we get an agreement between the experimental and calculated NMR data with the coarse model that is comparable to all atom models. Or we can incorporate mutational data, thermodynamic, thermodynamic changes upon mutation, like difference in stability upon mutation. If you mutate from this residue into this residue, we consider it as a perturbation. We use perturbation theory and this maximum likelihood approach. And we have tested it on different proteins. And again, we find that if we don't use, if we use functional form with only two-body interactions, we cannot find a model that reproduces the experimental data. If you use only two-body terms in the coarse-grained model, there is no model exists, no set of parameters exists that can reproduce the experimental data. You see some of them, so these are the experimental free energy differences upon mutation, difference in stability. These are the calculated, and this is the beginning, and this is the optimized, and there is always a difference. This, in particular, there is a set of mutational data that cannot be reproduced. However, as soon as we introduce a multibody term, we can fit the model very well. This is the initial model, we train the model, and then we, the optimized model, the red is this hybrid with the multibody interactions. We can reproduce experimental data. So again, this underscores the importance of multibody term and coarse graining, and again underscores the importance of machine learning in trying to model this system and the coarse resolution, because at the moment it's very difficult to come up with the functional form for higher body interactions. And with this, I would like to thank all the people who have done the work. In particular, my student Nick has been working a lot on this machine learned force field together with my postdoc Zhang. And all this work is done in collaboration with the group of Frank Noe in Berlin. Thank you very much.
Thank you so much, Cecilia, for the great talk. We're taking questions through the Q&A. We'll give everyone just a moment. All right, so first question is wondering if that would also be the case for classical force fields when trying to reproduce quantum effects. So in that case, where different atom species are introduced, would that be a viable option for your approach? Yeah, there are a lot of people doing this, doing a classical force field with quantum mechanical data. There is, for instance, there are several groups doing that with great success. It works very well. And actually, when you do it with classical force field, it's been shown that in the multibody expansion, it doesn't decay so slowly, because it's a different, the nature of the multibody interactions is different. In the when you go from quantum to classical, is the nature, the quantum mechanical nature of the electrons that that they are delocalized, that that give rise to multibody terms. While when you go from atomistic to coarse-grained, is the fact that you are doing this integral, this renormalization that I showed. This, let me go. This integral here
this integral here that mathematically gives rise to multi-body term. So the framing is the same. The results are a bit different. But yes, it's true that there are many people and using machine learning, all sorts of machine learning, not just deep neural network, for defining classical force fields from quantum mechanics, and they are extremely successful.
Great. We also have a follow-up question. If you could comment on the interference time of deep learning models.
So it depends on the architecture. Depends on how many systems you're training, depends on the size of the system, especially if you do it on a transferable architecture. So also depends, for instance, if you do this multi-body expansion, it depends how many term you put in the expansion. The inference is slower than the simulation itself. So it takes a while to train the network. It may take, for a very complicated architecture, right now we are putting like thousands of proteins together, that may take a week to train the model. But then the simulation is very fast. It is something you do once, and then you reuse it.
How important could multi-body interactions be for atomic force fields? And a little follow-up is, how come the 5-body energy plots look closer to AA free energy maps than the CGNet?
So, as I think I already answered the first question, people working on the multi-body interactions for atomistic force fields find that they are indeed important. For instance, there is the work of Francesco Paesani from UCSD, was shown that for water, 3-body corrections are very important, but already he has done 3-body, 4-body, 5-body correction. Actually, this work started because of some discussions with Francesco Paesani we had at IPAM in Los Angeles last year. And but they're not so, the expansion doesn't decay so slowly. The 3-body is the important correction. Then already the 4-body is very small, the correction. The 5-body is basically non-existent. And the reason is this different nature, as I said. Going from quantum to classical is the quantum mechanical nature of the electrons. And you need 3-body interactions to represent the interaction. But if you do a much more aggressive coarsening, and if you apply this normalization here, then you have other interactions appearing. And why the 5-body energy plot looks closer, I think is because of the normalization. So one problem with neural network is there is the risk always of over-parameterizing the model. You have many parameters. And if you impose some structure, you regularize it. So this is a sort of regularization, indirect regularization of the model that we obtain with, for instance, when we impose the transferability, when we impose the multi-body expansion, all the structure, additional structure and physical constraints that we put into the model act as regularization.
To zoom out a little bit, what are some examples of the larger systems you're working to accurately simulate? Like, would this also include things like protein complexes?
Eventually, we would love to model protein complexes. We're not there yet. So right now, we are working on a very large set of peptides. So we have some 2,000 peptides. We train all different sequences. We train on half, and we predict the other half. And this seems to work quite well. So it's really transferable. And now we are trying to apply the same thing to proteins, like single domain proteins, about 100 residue long. We take very large, we take the CATH database where we have the different sequences with minimum overlap of minimum similarity. And we train on a number of them. And then we want to reproduce, for instance, that these show folding simulation with the coarse-grained model, but this is in progress.
Can you comment on the cost of CGNet compared to traditional CG models like Go models, Martini, et cetera?
the cost, yeah, the cost is not yet as a Go model or Go model is much faster than Martini model, but the cost is not yet at the level of a Go model. Is much cheaper, is much faster than Olato model, a coarse-grained model with CGNet, but we haven't yet done anything to optimize for speed. Right now, we're only working on making it to see if it's possible. We are beyond the proof of principle, I think, but we haven't done anything on optimizing for speed. Now I've gotten a couple of new postdocs. We may start to think about making things faster, but we need to think about the architecture, the interface with molecular dynamic packages, because all of this is done PyTorch. The neural network park is done with PyTorch, and so we need to interface PyTorch with molecular dynamic packages. There are some tools to do that, but we haven't worked on the optimization of this. So I would say right now is slower than CG model, maybe a factor of 2 or 3. Some depends on the architecture, maybe even more, maybe even slower. But we need to start to optimize. So I cannot say how we can push it.
So the free energy surfaces you show are very well captured by your machine learning approach, but in the training sets, there's appears to be very little information about transitions between states. So eventually the mechanism, the inferred model, may not resemble the true mechanism. Do you recover correct information about transition paths from the machine learning force fields?
This is a great question. So the thermodynamic consistency doesn't say anything about the kinetics. One thing, the mechanism should be the same because the free energy, the minimum free energy paths are the same. If the free energy really matches, and we are not integrating out in slow degrees of freedom, this is very important. If we integrate out the degrees of freedom that is very slow, then we mess up the kinetics completely. But if we are careful and we don't integrate out slow degrees of freedom, then the minimum free energy path in the two projection is the same. So the mechanism should be the same, but the rates, no. We cannot say anything about the rates. In fact, we have started to work on what we call spectral matching, where we match the eigenfunction function of the dynamical operator to try to match the kinetics. But this is the beginning.
Similarly focused on training data, is there enough transferability between the types of training data sets that where you could say to train something using only a beta sheet protein and reproduce data of alpha helical proteins? How much transference do you get, or is it very specific?
This is what we're working on right now. So I think what we see is that at least with the peptides, what we see is the model learns the interaction environment. We haven't tested now we are testing on secondary structure. We hope the model is able to reproduce secondary structure. If we give some example, for instance, of one helix and a beta sheet in the training set, should be able to reproduce different kinds of different sequences forming beta sheets of alpha helices, different from the training, because the model learns the environment. But we are testing it now. I don't know what we hope that this is what we'll get, but on peptides it's worked very well. We don't know on proteins yet.
To follow up on the protein complexes question, do you have any data yet showing how well the CG energy net functions work for protein-protein interactions in contrast to just a single molecule, or?
We haven't done it yet. This is what we would like to do, but first we need to test it. I mean, we need to take step by step, because if it doesn't work, we don't know why it doesn't work. So we want to go systematically and increase the system.
Great. And we have somebody curious about if there's any prospect for including pH dependence in protein force fields.
Well, eventually, if we give examples at different pH, the network will learn different pH. Also, this force field that I've talked about with machine learning are all trained at a given thermodynamic condition, a given temperature, given pH, given the natural concentration, everything set. Even the temperature cannot be changed. So we simulate, all the system need to be at the same temperature because the network learns the parameters at that temperature. If we change, especially in coarse graining, if we change the temperature, the pH, the free energy, the effective energy parameters will change, because in the coarse grain model, the energy is actually a free energy we are using, not because we don't normalize degrees of freedom. So there is an entropy part that enters the energy, so it becomes a free energy. And so because of this, is temperature dependent and is depending on the thermodynamic condition or all the thermodynamic conditions. So all of this is at the set state, the set thermodynamic state, set temperature, pH, and so on. I think if this works, then we can give examples at different thermodynamic states, like different temperature, different pH, and the network should be able to learn this, how the parameters change. But again, this is work for the future.
Well, great. I know we've got, the audience is giving you a great grilling. Thank you so much, Cecilia. Let's take a moment and thank the speaker again. And if you enjoyed today's event, we're having our next distinguished speaker is next week on April 22nd. Miguel Besa, Assistant Professor of Material Science Engineering, TU Delft, will be speaking about machine learning and materials. And prior to closing up today, we'll just take a moment and I'll, Director Erica Klacek will come back and have a few closing remarks for us.
Thanks, Allyson. And thank you, Cecilia. I'm going to share screen here just to close up. There we go. All right, so yeah, definitely I'd like to ask you join me thanking Professor Clementi again. This was really a fantastic start to a series where what we really hoped to do was start bringing out this fascinating interface between deep understanding of structural theory-driven models and the incredible tidal wave of development that's been going on in deep learning, and understanding how researchers are beginning to really make new inroads in bringing those together. And I think this is a fantastic demonstration and tour de force of what's going on in this particular area. And as you see in the next few weeks, we're going to see how similar types of thinking is being done in material science, in atmospheric science, and ultimately in economics as well. I want to thank Allyson for hosting today. And if you would like to catch, if you missed some of this event or you'd like to rewatch or whatnot, the video for today's event will be posted on our YouTube channel. And then finally, on behalf of our junior faculty fellows who are our series organizers, I'd like to thank all of you for joining us. And I do hope that you can join us at future events. So thank you.